We're looking at clinical trials, which of course is absolutely crucial for so many, in so many different countries. I'd like to call on straight away Dr. Pascal Hamel from the Paul Brousse Hospital Villejuif, researcher also at INSERM. Designing clinical trials, how can we do that? Welcome, thank you very much.
I wait for the music.
Yes, of course. It puts us into the mood.
Yeah, thank you very much for inviting me, and a special thanks to this guy here, Norbert, who organized with all the Inca and UCMS. Very nice organization. Thank you. Well, the question is the problem to design trial for this very poor prognostic cancer. And this, of course, is important. That one, yeah. First problem with these trials is the limit of consensus for prognosis definition, poor prognosis definition of this cancer. Okay, or we will say quantitative, 5 years overall survival less than 30%, and incidence superior to mortality, almost like mortality. Also qualitative, you have resistant treatment, refractory, incurable disease. Okay for that. But the problem is only 5% of patients are enrolled in clinical trial. And for example, trial opened in 2011, only 15% reach their target. Of course, there is the greatest need for resectable tumor. We don't search a lot for resectable. We go right to the surgeon. It's a good idea for me to go to the surgeon, but nevertheless. And treatment in academic center and higher neighborhood education strongly influence the inclusion of the trials. For example, in that study, you have the reference down. Enrollment was over 20% in patients with high school degree, and it was less than 7% in no high school degree. So this is not very democratic. For example, here, patients in blue are black people, patients in red are white people, and you see that there are inequities in clinical trial enrollments and access denied. So we have to care about the population we treat, trying to be the most equal as possible. Of course, for pediatric patients, there are some difficulty. First, 2/3 of patients are in bad condition when we have to see them for the first time in consultation. There are metabolic reprogramming, immunity, circadian rhythm, microbiome change, and all these things increase the rate of patients when you can target that, you can have the possibility to increase the rates to target more patients for aggressive chemotherapy and surgical therapies. Then the problem, and we have to change, the classification are based on anatomical feature with our CT scan in multidisciplinary meeting and not on molecular feature for the moment. Okay, it's finished. Thank you very much. And we lack of predictive biomarker to select our patient for a specific treatment. And finally, I think that at the beginning of this research, I found that there were real reluctance of pharma to embark in some trials for this cancer, particularly for patients in a bad condition. We all focus on patients with PS01, okay, but most patients are PS2, 3 or more, and there are not enough trials for them. Again, 80 to 90% of novel treatment strategies in clinical testing don't meet criteria for regulatory approval. So a lot of failure. Majority of failure, final phase of clinical testing, we will see that. Which is important that most negative phase 3 trial, previously phase, have already shown these trends before we embark in the study. So there is a problem. We have to select more and more precautious phase 3 trials. And maybe it's better to fail earlier and faster to identify and describe this approach rather than go front in a phase 3 trial with a lot of patients and to be sure failure for the future. We have to increase the stringency for quality and depth of testing in preclinical study and measure of efficacy in earlier phases. This is not enough down. We have to increase information about presumptive targets and define more carefully go and no-go criteria before clinical testing. And finally, we have to work more in the future on tissue-based assay before but also after the treatment. And we know that post-surgical resected specimen offer more tissue than a little sheets of cancer cells on AUS FNR biopsy. For example, in ASCO 2007, I don't know, Jérôme, if you were there. It's probably the bad day of my scientific life. We presented a study. We have 4 randomized phase 3 studies in that single meeting. It was a plenary session of the ASCO. All 4 studies were negative. No question during the 4th presentation. No question. Silence. And more than 2,000 patients included in those studies. So probably the selection of this study was no good. More than 2,000 patients for nothing. And now I think we have to progress. The current paradigm is you make a little preclinical testing. Yes, we have probably a drug. Go in phase 1 with no defined criteria. Then go only safety. Safety is okay. Go embark in phase 2. And then search for clinical signal. And then finally, the Graal, go to phase 3 to try to be better than the standard of care or placebo. But I think the next paradigm would be to make a more strong and accurate clinical testing, test in validated platform, restrict go and no-go criteria. If you don't have that in preclinical testing, stop. And if you embark in phase 1, okay for the safety, but also is based on imaging-based efficacy to see if there is no signal, stop. And after phase 2, further safety, high bar for efficacy, and then okay for superiority over placebo. For methodology, there are some problems with crossover. We are testing now the Kairos inhibitor with revolution medicine drug. There is no possibility for crossover, and it's not easy for patients who are randomized for the control arm. So maybe you can make biopsy, treatment A, biopsy, treatment B, and rebiopsy to see if the tumor change. Of course, if possible, it would be nice to make a cross-evaluation for patients and interim evaluation to see if possible to stop earlier the study than planned. And of course, this is very important to try to improve, but also the comfort of the patients. But the problem is that if you make a crossover and the pharma are reluctant to that, you can have with one arm a better result. For example, our study published with Talia Golan in the New England about olaparib as maintenance therapy in pancreatic cancer. The study was positive. PFS was better in blue with olaparib rather than in placebo. But all people say, what about overall survival? Crossover was not allowed, but a lot of physicians gave finally olaparib to the patient who progressed rapidly, and the OS was negative. So there's a problem or risk of crossover. We have to develop in the future the maintenance concept. The maintenance concept, you give FOLFIRINOX as first line of chemotherapy, then progression or toxicity, then second line, then progression, then third line if possible, but the number of patients decrease, of course. But I think there is another concept now developed for 10 years, particularly in France. You give toxic first-line polychemotherapy, and after several months, 4 months, for example, the toxicity for oxaliplatin with neuropathy occurs, and then you can stop and try to reintroduce later. So you have less toxicity, but you can keep the drug in the pocket to reintroduce later. And this was, it has been shown to be efficient, like, for example, these maintenance trials in PDAC, with pursuit of FOLFIRINOX versus maintenance 5-FU, omitting oxaliplatin and irinotecan after 4 months, you see there is no difference in survival because they reintroduce after the drugs when the tumor progresses. And for G-BRCA mutation, of course, we have a nice difference between PARP inhibitor olaparib and placebo. And reintroducing after progression FOLFIRINOX or even olaparib allow the patient to go better. But to prove the efficacy of drug, you need these trials. I think what is lacking now, the care of patient, I mean the care around chemotherapy or surgery. We have designed this study within the year 10 years ago. It was a long time because to find some money to put this, it's very difficult because there was no pharma to help, no drugs. And we test usual care versus usual care plus 4 weeks of physical activity with a coach. And because we know that physical activity have a very nice influence of immunity against cancer. And we perform they perform aerobic training and also resistance to increase the muscle mass. And finally, blue curves, in all type of quality of life item, there were improvement by performing physical activity. More intensive than in a control arm, and partial response was obtained in more than 10%, and there were the trends to increase survival, but the study was not designed to see survival. I think it's very important to take that into account now. Another type of thing to take into account, in patients with PS2, but more too tight to go in our trials, we perform a 14 days integrative supportive care in those patients who were symptomatic and tired. Finally, improvement was seen in 23% of patients. They could start a chemo in 73%, and even they can receive polychemotherapy after 1 month of preparation of rehabilitation. So we have to care on these patients who are not fit to go to enter frontally a study. Finally, of course, we have to design those markers. It's a clin d'œil to Nelson and group, because probably we will decrease the toxicity of the chemotherapy with such predictive marker. And in the future, we have to screen, for example, large population with diabetes. We talk of that together during the lunch. And if you see 3 million of new diabetes in France every year, you cannot make 3 million of CT scan, okay? So we have to find a risk factor and then to analyze how to select patients who should have the better screening. For example, new onset diabetes plus pathogenic variant plus US positive, and then go to cancer therapy or surgery. But of course, large screening is not possible in diabetic patients for that. And finally, we will have help, of course, with endoscopic ultrasound and, of course, artificial intelligence, try to push algorithms to help us to choose the patient and select patients in the large population of diabetes patients. I think it's very important to manage and to prevent the deception of patients for study. You have, for example, in France, a bit late, I make fast, 16,000 patients with PDAC, new cases. A certain subset would have a BRCA mutation, but some progress during chemotherapy before proposing olaparib, some progress with olaparib, and finally, only half of patients have a good median PFS of 7 months or more. And with KRAS mutation G12C, G12C, you have only 250 patients in France every year, theoretically. They are not all eligible to chemo. Some progress during chemo, and some progress at the very beginning of the targeted therapy. And finally, the same for other rare mutations. Finally, to my opinion, less than 500 patients really benefit of targeted therapy. So it's good to try to target therapy, but it's better to find other solutions. So when managing this patient, I think it's very important to learn to young physicians to say, that situation, your tumor is borderline 1, we must start new chemo, and then how you operated you. That our surgeon says that very often. And finally, we find the patient after the neoadjuvant treatment and the tumor had progressed. And the patient there, and my operation, it's no longer the solution, and the patient is at this point. Exactly the same for the anti-KRAS protocol. We randomize between chemo and the new drugs, daraxonrasib. And I'm happy to you are eligible for this protocol. You can enter the protocol. Finally, you have been randomized in the control arm with chemotherapy. The patients are very disappointed. They cry. They cry in the consultation room. Okay. Could I receive the new drug afterward if the chemo doesn't work? No, it's not possible. There is no crossover. I think we have to think of that when we receive the patient and explain, because our new drugs, we are very proud and very happy with that, but half of patients will not receive the drug, and we have to train them to be more comfortable. There are a lot of anticipating problems, fear to be a guinea pig for patients, distance delay for inclusion, accessibility trial, inclusion-exclusion criteria, side effects, and caregiver and deception. And we have to fight all these aspects of a problem for anticipation. So finally, about one word about philosophy of trial. We all think with patient to survival, but we, doctor, think about, oh, more work if I include this patient is a lot of work for me, and I hope he will be included because I will find the best treatment in the world. But the patient don't think of that. He think of quality of life, which is probably very important in bad prognostic cancer because probably the life is short. So finally, it's my last slide. New therapeutic, yes, but also optimize older ones and test new strategy. Predictive marker of efficacy and failure are very important to determine. Favor limited, effective, randomized phase 2 trial before embarking in large trial to get approval. The importance of supportive care, insist again. Focus study on frail and aged patients are really necessary now for poor prognostic cancer, and maintenance trial for this cancer that can be controlled longer. We have to think about that. Thank you very much.
Thank you very much, Dr. Hamel, if you'd like to take a seat. Thank you very much. Lots of food for thought, and great to see the real conversations, examples of the real conversations on the human level. Thank you so much. Let's now talk about registry-based clinical trials. I'd like to call on Dr. Belinda Lee, who is an oncologist at the Peter MacCallum Cancer Centre, Northern Health in Australia. Welcome.
Thank you to the organising committee for inviting me to speak to you today about opportunities and challenges of registry-based clinical trials in pancreatic cancer. So as we heard from many of our speakers today, pancreatic cancer is a critical public health and growing service issue both in developed and developing countries. The WHO has already said that pancreatic cancer is the third leading cause of cancer-related death in the United States and several European countries. In the majority of cases, these patients, unfortunately, do not have long-term survival, and this can only be achieved through surgical resection. But unfortunately, only 1 in 5 patients is really suitable for surgical resection, and even after surgical resection, 80% of our patients will still relapse within 2 years. So the routine use of adjuvant chemotherapy has improved survival outcomes, but there are currently no biomarkers that define patients are more or less likely to benefit from this treatment. And we still have very limited therapeutic options, with only a 5-year survival that has improved, but is still limited to 13% approximately. So despite the growing volume of knowledge in pancreatic cancer tumor biology, the reality is that pancreatic cancer is very hard to detect and presents with very advanced disease, and we have very few therapeutic options. This is the timeline of drugs over the last 25 years. And what you see in black are drugs that are reimbursed by Australian Pharmaceutical Benefit System, and in red are the drugs that are FDA and TGA approved but not reimbursed for our patients. So what is the solution here? We need to understand a pancreatic cancer landscape. We need better early detection and better therapeutic options, as has been said by many of the speakers today already. So in 2016, I established the Purple Translational Registry. And at the time, we were hopeful that maybe 10 cancer centers would work with us. I'm pleased to say that now we have over 53 cancer centers working with us across Australia, New Zealand, and Singapore. And the idea of the registry is to support clinical trials, basic and translational research. But the goals are to consolidate data and research into a unique translational registry platform to improve data sharing and collaboration across laboratories and cancer centers, and to empower our patients and enable research. So we are the only integrated translational pancreatic cancer center platform in Australia. And the idea is that we retrieve clinical data, we map the data, and we support and accelerate research in pancreatic cancer. But I think most importantly, this is about interoperable, findable data. And this becomes increasingly important as our clinical trials become more niche. So we have also got a number of registry-based trials that run off the pancreas registry platform. This is the Dynamic Pancreas and the Direct Inspire study that I'll talk about briefly later. So here on the left-hand side is what we call a butterfly plot. And the idea is that the Purple Registry is about real-world data in real time. So we can see here the age distribution as well as the distribution by stage of patients as they present. And as we know that there's only a small proportion with resectable cancer at initial diagnosis. But I'm an optimist, and I looked at this and thought, well, actually, there's a lot more patients with localized disease than I expected, and maybe one day we will be able to downstage these patients to resection. In purple, the patients are metastatic. So this year, we've taken it upon ourselves to create a database which actually has all our clinical trials across Victoria listed. And this is really born out of my own frustration about finding the right trial for the right patient. So we're now expanding this across Australia and hopefully into Singapore and New Zealand if they want to participate. And the issue is that there are databases that have the clinical trials listed, but most of the time, this data is not accurate. It doesn't tell us which studies are actually actively recruiting patients and will they accept our patients. And so we are actually updating this every month so the data is accurate. And this is just a snapshot from the last month of the data trials, clinical trials that are running across Victoria. And here we see the targets as well as the proportion of the studies with those targets. And I think this is just interesting from a researcher's perspective because it helps us understand what the pharmaceutical companies are focusing on and helps us as researchers to maybe to focus our research as well. And we're doing this every month, so we will actually be able to see the evolution of the different targets and where the interest lies. So I'm here to talk to you about registry-based clinical trials. So we all know that randomized controlled trials are our gold standard. They have good internal validity and they reduce bias by their design. But we also know that these studies are very expensive to run, they take longer to recruit, and they have high complexity. We also have to go through very long regulatory processes, and often there's a select population which may not apply to the general population once the drug becomes available to everybody. On the other hand, we have clinical registries, which are great. They provide external validity, they're real-world population, we can identify patients through the registry, and we have existing data already. But we also know that there's inherent bias in registries and that there's less stringent control and monitoring, so the data is not as clean. But if we can combine the two to what we call a randomized registry-based trial, then we get the best of both worlds. We get the gold standard of a randomized controlled trial, but we embed this into a registry. And this means that we can run the studies faster, it's more pragmatic design, and we already have the follow-up because we're already following those patients. All we have to do is build new modules for each trial. We also get broader data linkage for these patients as well. So this is an example of one of our registry-based trials. It's called Direct Inspire. It's quite a niche study. It's for locally advanced unresectable pancreatic cancer patients who've had chemotherapy, have not been able to go forward to their surgery, but instead we can offer them nanoknife or irreversible electroporation. So it is quite a niche because the centers must have the nanoknife technology to be able to run the study. But the idea is that with these registry-based trials, we can answer questions for rarer cohorts or where the events are less common. It's also faster to adopt the evidence in practice. And we can start looking at some of these questions that we talked about, looking about questions about optimal efficacy or combinations and non-inferiority questions. These are questions that often the industry pharma companies are not particularly interested in answering these questions. They're cheaper to run because the registry already exists. And it also means that smaller cancer centers can participate in the study if they don't have those big trial units to help them run studies. It reduces the time taken also to create the specific modules for the trial. And then there's the other issue of the patients and their ECOG status. So in a registry-based trial, patients can be older, they can have higher ECOG status, and we can accept patients who've got real-world comorbidities as well, which are often out-selected from other clinical trials. And then we also link our data to the National Death Registry, so we have got the long-term follow-up. I'm going to move on to talk about how registry-based trials can be integrated into translational research. So this is data that was presented last year at ASCO from our dynamic pancreas trial, and this is a trial that investigated the use of circulating tumor DNA to help guide adjuvant chemotherapy treatment. So the study is run across 26 cancer centers, and we had 102 patients enrolling in the study. So patients enrolled after their curative intent surgery, and they had to be fit for adjuvant chemotherapy. They were then stratified based on which chemotherapy their treating clinician opted to give them. So you could have a gem doublet with gemcitabine capecitabine or modified FOLFIRINOX. It was non-randomized for this particular arm of the study. So all patients had ctDNA-informed results. If they were ctDNA positive, they would receive 6 months of full adjuvant chemotherapy. And if they're ctDNA negative, they could be de-escalated to 3 to 4 months. But this was optional based on the physician and the patient's approval first. So what we found from the study is that real-time ctDNA analysis is feasible. We're able to turn around the ctDNA collection with the results within 5 weeks, and all patients started their adjuvant chemotherapy within 6 weeks. We also noticed that 41% of our patients after curative intent surgery were actually ctDNA positive. When we asked the clinicians and the patients what type of chemotherapy they wanted to receive, 65% opted for modified FOLFIRINOX, and 35% chose gemcitabine and capecitabine. So at the initial planning stage, 55% of the clinicians said that if their patient came back with a ctDNA negative result, they would de-escalate. And if they're ctDNA positive, they would give them the full 6 months of chemotherapy. But what we actually found at the end of the study was that, in fact, within the ctDNA negative cohort, 67% of patients received less than 4 months. And the reason for this is often patient preference to stop the chemotherapy early and sometimes due to toxicity. But also concerningly then in the ctDNA positive cohort, who is supposed to have received six months of chemotherapy, and all of them, actually 50% of patients received less than 6 months. But here the reason for stopping early were mostly due to toxicity in 33% and relapse in 18%. So we can confirm that ctDNA analysis is a prognostic biomarker in early-stage pancreatic cancer. At a median follow-up of 36 months, we can see that those who are ctDNA negative had a recurrence-free survival of 22 months compared to 13 months in ctDNA positive cohort, and this was statistically significant. In the interest of time, I'm only going to talk to you today about the ctDNA negative cohort. So in our study, we wanted to compare duration of chemotherapy, and we also want to look at the intensity of chemotherapy. So on the left-hand side, within the ctDNA negative cohort, we compared patients who had 6 months compared to 3 or 4 months of adjuvant chemotherapy. And even though there was a difference in recurrence-free survival, this was not statistically different. And then when we looked at intensity of chemotherapy comparing triplet therapy to doublet therapy, again, even though there was a difference in recurrence-free survival with 27 months with FOLFIRINOX and 18 months with GemCap, it was not statistically different. Perhaps we think there may be a trend here, though, with the modified FOLFIRINOX. So what were the learnings from dynamic pancreas? We can confirm that a tumor-informed ctDNA analysis following resection of early-stage pancreatic cancer has potential clinical utility. ctDNA is a significant prognostic marker in early-stage pancreatic cancer. Detection of ctDNA was associated with early recurrence, but high risk of recurrence persists even in our ctDNA negative patients. Gains in survival for early-stage pancreatic cancer are dependent on developing better strategies for optimizing adjuvant chemotherapy choices and better treatments. So I'm going to move on now to talk about the translational aspect of our registry, the idea of learning from every patient. And the idea is that with one patient, we can do multiple para-analysis. So at the moment, our pancreatic registry is also a biobank. So we're collecting tumor tissue, blood, for which we then spin down for PBMCs, plasma, and serum. So we've been doing multiplex IHC, cytoph, proteomics, and cytokines as well. I'm only going to speak to you about one of our projects in the interest of time. So here I'm presenting you some data on blood-based proteomics of the plasma. In this study, we enrolled 449 participants, and we did this in 2 cohorts, a discovery cohort and then a validation cohort. Within our discovery cohort, there are 200 over patients. We compared pancreatic cancer patients against healthy controls and patients who had lesions in the pancreas that were not pancreatic cancer. We then validated this against another cohort with pancreatic cancer and healthy controls, but this time around we included colorectal cancer. And the reason we did that was we wanted to know that the signature we're finding, was it truly to do with pancreatic cancer and not just because they had a GI malignancy. We extracted the clinical data from the Purple Translational Registry. We matched it against their treatments that patients received, their responses, and their overall survival. So what we were very pleased to see was that when we compared, this is hierarchical clustering plot looking at the differential expression of proteins, that there was a clean signature when we compared pancreatic cancer to healthy controls. We're very excited by this. We then did the same analysis comparing alternative lesions in the pancreas with healthy controls, and as expected, that was a clean signature as well. But most importantly, when we compared pancreatic cancer against those who had lesions in the pancreas, again, we could still see that there is a clean signature. And this is important because it means that your findings aren't just to do with inflammation within the pancreas. These are what we call log-log plots, and here allows us to start to identify the individual proteins and find those that are uniquely changing. So we compare late pancreatic cancer with colorectal cancer, and we identify those that are specific to pancreatic cancer. We do the same with the early pancreas cancer versus colorectal cancer. And finally, we want to look at those that are only up in early pancreatic cancer, because potentially these might be helpful for detection and earlier screening. So this has been a very quick talk. I've got 1 minute left. And really, I just want to be able to say, look, this is just giving you food for thought for how we can run registry-based trials. I want to emphasize how important it is to collaborate, that by collaborating and having this type of infrastructure, we can do faster research. And potentially, we're hoping to do more adaptive registry-based trials into the future. I think we need to learn from every patient. There's a lot of people to thank for this. There's 53 cancer centers that are working with us, and I'd thank you to all of them. I'd also like to thank all the laboratories that we've been working with, all our various different studies, and to all the patients and the carers and our funders. Thank you.
Thank you very much. Indeed, learning from every patient, a nice phrase to have at the end there. Let's now get the perspective from the chair of the European Study Group for Pancreatic Cancer, Professor John Neoptolemus, University of Heidelberg, but from London as well. John, welcome.
Thank you, Seamus. I'd like to thank Juan Ivana for organising this really superb meeting. My talk has really been moderately, to a large extent, already given by Dieter and by Steve Gallinger, to whom I'm very grateful. Now, which button is it?
The big green button.
Oh, the green one, yeah. Okay, so straightforward stuff. Surgery is really important. We're talking about surgical trials. We need to contrast T&M staging with empirical staging. We need to contrast the Hallstadian concepts of cancer evolution versus biological evolution. And this is my talk, really, that I'm going to give. Cancer is not a fixed entity in any one patient. And so there's something called plasticity, which is evolution, change, and so on. And it's driven by clonal evolution over time and space and intervention. Any intervention we make will change the phenotype and more of that cancer, epigenetic even. So how do we improve treatment outcomes? Well, we've got to acquire the tissue, because this is what kills the patient. And unless we treat that tissue in the right kind of way, the patient will die. And of course, we need different types of investigation to be able to fully understand what we're dealing with. So these are the big killers. We all know lung in men, prostate, colon, pancreas, liver. Women, pretty much the same. Add breast, add uterine cancer too. So there are important trends with the big cancer killers over time. These are American data. Lung is decreasing, mainly due to reduction in smoking. Some progress has been made with treating breast cancers and colorectal cancer. But you can see, if you move over to 2020, that survival rates are actually solidifying. So in terms of what we've been doing, we've sort of reached the glass ceiling. So how do we get through the glass ceiling? So this is Halsted, revolutionary surgeon in his time. What was revolutionary about it? Well, the Greeks and the Romans were already removing breasts for cancer. They knew what cancer was. And they weren't bad surgeons, you know. And they had to do it with general anaesthetic and no proper hemostasis. So he came up with the idea, breast cancer keeps coming back again, so we have to chop away more and more of the surrounding tissues, the Hallstadian concept of sequential centrifugal spread. And it got to a stage where actually they were removing rib cages and all the rest of it. Never really worked. And so Steve Gallinger summarized this up in a review paper in 2022, where he contrasted Hallstadian spread. It goes to the lymph nodes, and then it goes to the lymphatics, and then it goes somewhere else, and then the lungs, and lungs to bone, and so on, with a sort of genomic-based evolution. Schizophrenic, because the two don't actually overlap. And I spoke to him earlier and he admitted that. So I trained at Guy's Hospital. I went to Cambridge University and then I went to Guy's Hospital in London because I was a London boy, you see. And one of my tutors actually was this guy, Hedley Atkins, and he's a nice guy, huge private practice in Harley Street. But he had time to do a randomized study where he decided to roll back the idea that we should be removing women's hemithorax to achieve cancer control. And he showed that in the early stages, yes, there wasn't a big difference between really big surgery and survival. The message that went out to all the oncologists and the radiotherapists and so on was the surgery is not important. That's what you're left with. But actually that's not true, because if you look at the more common, more advanced stages, surgery makes a big difference. So surgery is important. And we've seen the same lesson. This time Bill Hild, a non-academic general district surgeon working in Basingstoke, showed that if you properly remove a rectal cancer, and these are rectal cancers here, that if you properly remove a rectal cancer, you get better survival. So surgery matters. If we look at lung, we see that early surgery gives you the best survival, over 60%. Our problem lies with the ones that aren't removed early, and some very, very nice studies, particularly coming out from Charlie Swanton and other colleagues at the Watson Crick Centre in London, have been able to track the clonal evolution of cancers in a spectacular manner. And I mean, the only cancer type, I think, that has been so studied in this way. But Steve Gallinger earlier on did mention, sort of anecdotally, the same kind of thing when he was talking about pancreas cancer. And that metastasis is actually part of the evolution that's taken within the primary and setting up the set for spread. So pancreas cancer, this is my great love. Why did I fall in love with pancreas cancer? Because all my cancer patients with pancreas cancer were dying. And unlike my other colleagues who were killing them on the operating table, I wasn't, and they were still dying. And so this is it's painful for a surgeon to lose patients, even if it is 12 or 18 months down the road, and having to deal with their families, their children or their wives or their husbands or fathers and mothers even. So, so surgery has developed big time in pancreas cancer now, and it's now possible to resect even the most locally advanced pancreas cancer in a way that we could never do before. And so this is go back. So these are locally advanced pancreas cancers. They survive. They don't survive forever, but they survive compared to no resection. And these are survivors in the SPAC4 adjuvant trial, and you can see that you're reaching, even with 108 month survival rates, you're achieving survival rates of up to 50%. This is real. These patients are alive. They come and kiss me in the street sometimes if they see me, saying, "You operated on me 20 years ago." I can't remember who they were, but it happens. And the other thing that has sort of changed our thinking, Raoul mentioned epistemological change. And so this is the other thing that's happened. We've stopped thinking along TNM, which is a reductionist approach, to an empirical approach, which is it resectable? Is it borderline resectable? Is it locally unresectable? Oligometastatic, polymetastatic? This isn't TNM, but it's what is going on at the moment. And curiously enough, it does actually reflect more in terms of what we do, and almost certainly is closer to the true biology of the cancer than we had previously imagined. And what you do in these kind of situations changes adjuvant versus neoadjuvant versus induction versus selective induction versus palliative standard of care. So these are the 5-year survival rates of the bad cancers. If you look at pancreas, we're doing okay with the localized resections. Here, sorry, go back. Not bad. Huge improvement. Huge improvement. When I was training, everyone was dead by 9 months, surgery or no surgery. So this is a big, big change. But we still got a problem here, and the ones that we can't get to surgery, and of course this is the majority of the patients and one that we need to focus on. A curiosity is, this is why. This is my trial, 2001, Lancet, and then oncology, surgery plus adjuvant chemotherapy, not radiotherapy. Nobody mentioned this, especially if you're in America. You don't want to mention SPAC. Why? Don't know why. It's simple, it's clear. And then SPAC 3 and 4. Pascal Hamel, where are you? Predict 24. You know, the French confirmed it. You know, the French always follow the British, you know. But what can you say? They have such beautiful architecture to perform. They don't want to really get into a dirty fight, so they give up. But then they support us. So that is the big difference. But we have to move on now, right? And so this is from Steve Cullinger's group in Toronto. The single genes don't change, but the genetic characteristic of the tumor does change. So we're moving from, in the transcriptomic situation, from a more classical to a more basal type. Tetraploid is very important. In other words, the increased number of chromosomes that you have. The tumor burden, the mutational burden increases, and the nature of the mutant KRASs also increases. Another important point to understand is that the transcriptome doesn't particularly follow TNM. It follows its own kind of classification, which is, again, another reason why not to think that a reductionist TNM approach should guide what we do as clinicians. The pathologist shouldn't be telling our surgeons or oncologists what we should be doing. We should be asking the pathologist for more information, not to dictate treatment. And so if you give someone chemotherapy, you get this change. So this is the Grunwald classification of reactive, intermediate, and deserted stroma. And with chemotherapy, you move to a reactive, which is a good thing to have, to a deserted stroma, which is a bad thing to have, and with a decreasing survival rate. So our intervention makes things worse. Hello? Yeah. So this is what we're talking about: plasticity, changes over time, changes with intervention. And this is what defined the approach for the SPAC 6 and 7 clinical trials. So it's a clinical trial which combines an awful lot of tissue collection, genomics, transcriptomics, but also acquisition of organoids. In other words, these are tissues that are derived from the actual patient themselves, both epithelial cells, the cancer cells, and the CAFs. I won't go into it, but you can go into functional analysis to understand better what is going on in that particular patient. And of course, artificial intelligence will come into this somewhere. But if it's done within the context of a randomized trial, then you have a better handle on what you're actually achieving, as opposed to what you think you're achieving. This is real world stuff. Right, so this is SPAC 6. We're about to start recruitment very shortly. It's quite simple. SPAC 6 is for resectable disease. It looks complicated. It's not. Patients are randomized into a standard arm, which is FOLFIRINOX. The experimental arm is randomized into oxaliplatin-based therapy or gemcitabine-based therapy. And this is based on an RNA-seq determined transcriptomic signature. It's nothing like the signature we heard this morning. This is the SPAC signature. And this has been developed only on human tissues and only on human survivals, not on PDXs, not on organoids, not on primary cell cultures, not on AI paraffin sections, only on human tissue. And it follows strict quality management systems and control and algorithms which are built in. And attached to this is the translational programs. SPAC 7 is similar, but in this case it's for locally advanced, and these patients get induction chemotherapy. And the signature again is the same as the RNA-seq signature, except we use nanostring to achieve a quicker turnaround for the randomization. So really that's all I have to say, apart from saying thank you to sites that are contracted. Germans love their contracts. We have contracts with these centres around Europe. And so thank you to everybody.
Thank you very much. Thank you very much. Maybe a right of reply from France to start with. I don't know if we need a right of reply. Listen, we have time for a couple of questions from the audience. Yes, we have hands straight away up for our three panelists.
Thank you, everybody, for that was a really great session. My question is for Dr. Lee. In terms of registry-based randomized trial, number one, do you get a sense that patients stay on in these trials longer for longer-term follow-up type approaches like you showed? And the second question is, one of the challenges we often see is that the control arm changes in the midst of a trial because some other treatment gets approved or whatever. And is that more facile to deal with in a registry trial, or it doesn't matter?
I mean, I think the first thing to say is, registry-based trials, great idea, and we're still working on it. They're actually not as easy to design as everyone thinks, but the idea is about stacking the data, that we don't lose data from other trials. So we just want to keep the data, so that in itself makes it part of a registry trial. We're not losing too many patients from the trial, so we are allowed to track them the whole way through their treatment. So I can't remember the second part of your question.
The second question was when the control arm changes in the midst of a trial. I mean, this has been a real challenge with many trials, right? I mean, you start with a monotherapy and then there's a combination that gets approved that's better. And then now you've ended up halfway through a trial with something where
I think we haven't quite. I think that is a real problem that that we will face. We haven't quite got there yet, but I think yes, we're going to have to look more into that. But at the moment, it hasn't been an issue so far.
Any other question from the audience? I'm also watching questions from people online, but nothing at the moment. Any other questions? Maybe also, does anybody want to react among the panelists from what we've heard from somebody else's presentation? Pascal, what are your thoughts after listening to your colleagues when it comes to clinical trials and so on? It's a hornet's nest.
It's a difficult question. Of course, I completely agree with what we was said before, because my topic was to imagine something more in the future, and this is the present. I think my colleague, she's the present now, and there are a lot of difficulty to finish the trials. I don't know if there is there are some difference between countries. I know that probably in the US it's sometimes more difficult to go on a randomized trial. In Europe it's not so difficult, but I think again the power of convincing of clinician, physician it's very important. What's about Australia for that?
Yeah, and Belinda, but also taking in New Zealand, Singapore, as well as Australia.
I think I guess so talking to one of our other colleagues about this, I think sometimes with the randomization, I think Australia, we don't have too many issues with it. I do suspect, and this is my own personal opinion, that sometimes funding and the way countries had the funding can have influence on how patients agree to or not agree to be on clinical trials. Because in certain countries we can get access to some of these drugs anyway, so therefore there's less incentive for them to go into clinical trials. I think between the different countries there is difference between Singapore, New Zealand and Australia. I think New Zealand and Australia are probably more inclined to go into trials, because especially when they can't get access to the drugs.
Well, registry trials. Well, I think it's okay for data gathering. I think it's good for collecting tissues and blood and so on, and sort of improving your databases for hypothesis generating. But in terms of clinical trials, I mean, it's an oxymoron, surely, when you're dealing with poor prognosis patients, because, all the bad ones are dead after a few months of registration. So what's the point? There are so many biases involved. And we get this a lot with the National Cancer Data Registry in the United States. I review a lot of papers that come from there, and they're all biased and they're all rejected by me. So, I mean, because they're intrinsically biased. So I think you can use the registry studies as opposed to trials in the right kind of way. But to say this is the way to advance human understanding and knowledge of poor prognosis cancers, I think is a slight, misdirection.
Interesting points. I don't know, Belinda, if you wanted to reply to that, and also Pascal as well.
One question, what your opinion about synthetic arm? Because we are more or less reluctant to propose a placebo in many trials, particularly in poor prognosis cancer. Everybody is excited by the idea of synthetic arm, which is an artificial way to compare the drug that we believe in. And I think I believe it's more complicated that in theoretical way. What about your opinion about that?
I mean, I think I'd agree. We are very interested in doing that. We haven't done that yet. I think part of what we're doing is building that infrastructure to be able to analyze the data, to have it, because without having the registry-based data to start with, you can't even start asking the question about synthetic cohorts anyway. So you have to have the data to start with to be able to do that. That's the first thing. So I think I can't say whether it's better or not better. I think it's definitely an idea we're looking at, we're toying at. I think this is where all your artificial intelligence comes in, all your modeling comes in, but we're not quite there yet. And maybe there are really intelligent people in the audience here who would like to come forward and offer their help with that. That's something that we need to work towards. I don't think that registry-based trials are the answer to everything. I think the idea really is that we have a registry. We know that we're running clinical trials. We know that when those individual trials close, we lose that data. And the idea is that we're trying not to lose the data. So you can still, I'm not saying we don't run commercial studies, we're running a lot of commercial studies, we're running a lot of clinical trials, but the idea is we're trying to hold our data so that we can create this massive database of big data. Because only when you've got these massive big data, then you can start to do all your modeling, then you can start to ask artificial questions. So I don't think that every trial can be a registry-based trial. They're actually quite difficult to design, but I think it is just an idea. And I think it answers questions which, pharmaceutical companies are not going to want to answer. So if you talk about looking at maintenance therapy, yes, that's potentially a really good one for a registry-based trial. Or you want to look at just combinations, just tweaking combinations that we're already using. That's the kind of study that you can do in a registry-based trial. So I don't think that all trials will be run like this. I think it's just an idea of potentially some ways to do it, but to hold the data so we're not losing it.
And just before the coffee break, interesting topic, another panel we could have for a whole day or 2 days or 3 days just on this topic. Dr. Urrutia.
Yes, just trying to harmonize previous talks with this talk, right? So I've been a big proponent in the United States from our center to actually go with the trials and do genomic for ethnicity. In other words, truly arrays in order to know the percentage of ancestry that we have, rather than the self-reporting ancestry, which sometimes is 5% of what the patient actually report. Now, I understand that in some, and this is a G7, that's why we have to talk about it, because I understand that some places in some of our countries do not take into consideration ethnicity or ancestry in the clinical trials. And that is, if we are thinking about it like a group, right, it's in contradiction with what we were saying in the previous discussions, in which we do have to include, right, as much diversity as we can. So what opinion do you have? In your case, do you include ancestry? And what type of ancestry you include? Is self-reported or measured genetically?
A brief response before the coffee break, and it's something we can continue discussing, of course, during the coffee break as well. It could be quite technical. Belinda, a brief answer.
Well, firstly, we aren't doing the genomics on everybody, so no, we can't do that from that perspective on the ancestry. It's self-reported, but it's two ways. So one of them is self-reported, and one is some hospital records. So what's recorded in the hospital. So we ask questions about where they're born. We ask about the language they're speaking, particularly for Australia. We ask about Aboriginal origin as well. Yep.
Lots of different approaches, a great topic, obviously lots of viewpoints as well, so great. Let's continue the conversation during the coffee break. Let's take 10 minutes. When we come back, we talked about cross-border collaboration. That's our theme after the coffee break. Let's take 10 minutes until 16:40. Thank you very much to our panel. Thank you very much.